So we have some different viruses of different stages of maturity outside the cell.
Will attach to a receptor on the surface of one of your cells.
One of these viruses will attach to a receptor on the surface of the cell.
That binding of the viral particle, to the receptor on the surface of your cell enables the virus to get inside. So it goes inside through a process called receptor-mediated endocytosis, which basically means the receptor helps it.
It then triggers receptor-mediated endocytosis, so the virus enters into the cell.
To get across the membrane and into the cell. So it's called receptor-mediated endocytosis, and that's the mechanism by which dengue virus particles enter your cells. Okay. Once they're in your cells, they form what's called the endosomal vesicle.
When they're in the cell, they form an endosomal vesicle,
which is basically just the virus particle inside an endosome. And that virus particle will then undergo acidification. It's quite acidic in there. And that acidic environment will cause it to uncoat.
It then undergoes acidification within the endosomal vesicle because it's quite acidic, and it loses its coat.
So what that means is it throws the nucleocapsid out. So you've got your nucleocapsid,
and it just throws the nucleic acid out,
which is in your envelope, which is in your membrane. It's all nicely packaged. In order for the dengue virus's genome, I suppose, its genetic information to be, which basically means, here you go, you can see it here, the nucleocapsid, which remember that contains our RNA, our genome, the nucleocapsid is spat out of the rest of the viron, and it's sort of released into the cytoplasm of your cell. So here it's nicely sort of contained in an endosomal vesicle, but at this point the DNA, to the endoplasmic reticulum.
The nucleic acid with the RNA inside then moves to the rough endoplasmic reticulum.
Can go through several stages of translation and processing.
And goes through processing and translation.
So in step 6, what we have is the dengue virus RNA genetic information is replicated.
So you can see here the RNA, the genetic information of the virus, is replicated.
Nucleocapsid being released here, step 4. It moves over the 9 off. We've got one copy of our RNA here. The RNA is replicated. It then needs to be put back into a nucleocapsid.
It's then put back into a nucleocapsid.
So the RNA kind of replicated on the surface of the endoplasmic reticulum. It's put inside a capsid to form a nucleocapsid, which is what's happening here. And then those nucleocapsids go back onto the endoplasmic reticulum.
The nucleocapsids then enter back into the endoplasmic reticulum and
and are assembled into the virus particle.
And are assembled into the viron particle, as you can see here.
So at this point, all you've got is a bit of RNA inside a capsid, inside a protein. At this point, the assembly point, all those other genes within that RNA are translated, and the other proteins are...
All the genes in the RNA are then translated.
And it's assembled into a full virus particle,
And it's assembled into a full virus particle.
So we get all the other membranes coming out, which is what you've got here. See how it goes from just being a capsid, nucleocapsid, to being a full dengue virus particle. Make sense? Yeah, sort of, yeah. At this point, it leaves the endoplasmic reticulum and it goes through the Golgi network.
At this point, it then leaves the endoplasmic reticulum and moves through the trans-Golgi network.
The Golgi network has sort of an decreasing, I should say, pH. And through the different pHs, it basically leads to virus maturation. So
and as it moves through the different pHs, it matures.
Things happen to the proteins and it becomes mature. Here, and off it will go into your cells and it will go on and infect.
It's then released and off it can go and infect more cells.